Our last topic in this section on plant form and function is a little bit about hormones. We're going to be looking at what hormones are, how they act at the cellular level, and some examples of plant functions that are regulated by hormones. So although we are explicitly talking about plant hormones, the generalizations that I make about hormones also apply to animals as well. Okay, so first question, what are hormones? Hormones are chemical messengers. Chemicals are a major way that cells communicate with each other. There are several different kinds of chemical messengers, and hormones are one category of them. The chemicals here include proteins and peptides. It includes amino acids and molecules derived from amino acids. It includes lipids, and under the lipids, steroids are an important group of hormones, and also, particularly in plants, some alcohol-like chemicals. This slide features the structure of two steroids. An animal steroid called estrogen, that's the primary female sex hormone in animals, and a sort of plant analog, a phytoestrogen. All right, so hormones are chemical messengers. There are many different kinds of chemical messengers. Some work within a cell. Hormones are messenger molecules that work outside of one cell. They're made by one cell and transported to another by fluids. The fluids in plants would include the sap in xylem, the sap in phloem, generally any extracellular fluids. In animals, we have a very specific definition of hormones. Hormones are chemical messengers that travel through the bloodstream, right? So hormones are chemical messengers that are made by one cell, and they are transported or they move to other cells by way of fluids. So again, the general things I say about hormones apply to both plants and animals. This illustration is based on an animal model because that's a blood vessel. And we see a cell secreting hormone molecules into the bloodstream, and those hormone molecules transporting to its target cell. If that was a plant, it might be phloem. So our definition is building here. Hormones are chemical messengers. They travel through extracellular fluids, and they bind with receptors within or on the surface of target cells. The target cell for a hormone is the cell that bears the receptor for that hormone. Although hormones come in a variety of chemical types, receptors are universally proteins. If that protein is embedded in the plasma membrane, it's a surface receptor. That would be the case for hormones that are water-soluble and polar. If the receptor is internal, that would be the case for non-polar lipid-soluble hormones. They can cross right through the plasma membrane and bind to receptors on the inside of the cell. So that idea of surface and internal receptors is captured in this image. On the left, we have a water-soluble hormone, such as, let's say, a protein or an amino acid derivative. It is binding to a receptor and working in an indirect fashion. Whereas on the right, The lipid-soluble hormone, such as a steroid, will pass right through the plasma membrane, bind to internal receptors, enter the nucleus, and have a pretty direct effect on the target. So hormones are chemical messengers traveling through extracellular fluid, binding to receptors, and altering the physiology of the target cell. Now, there are several different ways in which hormones affect targets. And I'm not talking about specific changes in the organism's body. I mean, at the cellular level, there are several mechanisms by which hormones alter the physiology of their target cells. And the best description of this in your book can be found back in chapter 5. That was the chapter about membranes. So you should really review pages 114 to 119. I'll summarize it in the next few slides, but that's a good presentation about how messenger molecules like hormones act at the cell level. That's what I'm calling the mechanisms of action. So we're going to look at 5 different mechanisms by which hormones alter the physiology of their target cell. And the first is that the hormone can change the permeability of the target cell membrane to ions. This would be the case if the receptor for the hormone was a ligand-gated channel. Channels are membrane proteins that allow facilitated diffusion, and some channels are gated. Gated channels have to be stimulated to change shape and assume their open form. A ligand-gated channel is one where the binding of a molecule to the channel causes it to undergo a shape change. A ligand is any extracellular molecule that binds to a surface receptor. So in this figure, we see the ligand is the hormone. The binding of the hormone to the receptor causes a change in the shape, the tertiary structure of this protein. It assumes its open conformation. It allows ions to diffuse in the cell. That would alter the charge on the inside of that cell and result in some kind of cellular response. Our second mechanism is that the binding of the hormone to the receptor activates a series of membrane-bound molecules, G proteins and enzymes. So in this figure we see the signaling molecule is the hormone. It binds to its receptor. The receptor is a protein that in turn activates a molecule called a G protein. And there may actually be a complex of G proteins. So the binding of the hormone activates this G protein. In its activated form, the G protein moves, sidles over, and binds with an enzyme that's bound to the membrane that was previously in an inactive shape. And the binding of the G protein activates this enzyme. It starts catalyzing its target reaction, and that changes the physiology of that cell, causes a cellular response. Our third mechanism of hormone action is for the hormone to activate a signal transduction pathway within the target. This is similar to the G protein example, only the molecules, the intermediate molecules, are not membrane bound, only the receptor. This is based on an animal example because the hormone is epinephrine. The goal, I guess, if you could say here, is to get this cell to convert glycogen to glucose. And that is a reaction that's catalyzed by an enzyme. But the enzyme is currently in an inactive form. So we have to activate that enzyme. That's the job of the hormone. The hormone can't pass through the plasma membrane and do that job itself. It has to bind to a surface receptor. And the binding of the hormone to the surface receptor sets into motion a chain of reactions where proteins on the inside of the cell become active and in turn activate the next relay molecule in the chain until we finally get the last relay molecule active that's capable of binding to the protein, converting it into its active form, and allowing that change in the physiology of the target cell to happen. Mechanism number 4 is kind of a mashup of the last two. Our second mechanism involved G protein activation and the binding of a G protein to a membrane-bound enzyme. That's happening here. The example number 3 involved an internal molecule becoming active, a transduction pathway. And that's sort of happening here as well. Only the internal molecule that's activated is not called a transduction pathway relay. It is called a second messenger. Second messengers are non-proteins, whereas the signal transduction relay molecules are all proteins. All right, so this example, we have the binding of the hormone to the receptor. The receptor activates a membrane-bound G protein. The G protein moves over and binds to a membrane-bound enzyme called adenylate cyclase. And adenylate cyclase strips 2 phosphate groups off of ATP, turning it into a molecule called AMP, cyclic aminosine monophosphate. And the presence of AMP, the second messenger, causes otherwise inactive enzymes called protein kinases to become active, and that changes the physiology of the cell. Mechanisms 1 through 4 all involved hormones that were water soluble and therefore could not cross the plasma membrane and had to work less directly. Our last mechanism here happens with lipid-soluble hormones like steroids. They directly influence gene expression. So we have the hormone crossing the plasma membrane, binding to a receptor on the inside of the cell. That complex of hormone plus receptor binds directly to DNA. Each hormone receptor complex would have a target gene it would bind to, causing transcription to occur. mRNA is produced. That leaves the nucleus and translation occurs. So we directly have the production of new proteins. These steroids and other lipid-soluble hormones can lead to gene expression. Now we're going to look at some specific processes in plants that are regulated by hormones. And the first one is called phototropism. Tropisms are responses where plants orient toward, if it's a positive tropism, or away, if it's a negative tropism, from a stimulus. Plants are positively phototropic. The shoot system, the above ground parts of plants, orient, grow toward light. That's called positive phototropism. Now, in animals, we have a response called taxis. So in animals, we don't use the word tropism, we talk about taxic responses. So an animal that moved toward light would be positively phototaxic, but in plants we say they are positively phototropic. I'm sure you've witnessed this before. If you have a plant on a window ledge, it grows toward the window, right? That makes sense. Plants are photosynthetic. Growing toward the light is an adaptation. But how does that work? I mean, how does the plant know to do this? The history of phototropism goes back a long ways. Actually, Charles Darwin did experiments, simple observations to try to understand the nature of phototropism. He used oat seedlings. So in the control here, you can see how once it germinates, the cotyledon orients toward the light. And he found that if you cut the tip off, it doesn't do that. So, the cells at the tip are responsible for this. So he covered the tip with an opaque cap. It's like putting blinders on the seedlings. And sure enough, even if the cells are there, if they're covered from light, the plant loses the tropic response. If you cover it with a transparent cap, it retains that. So it's light hitting the cells at the tip. Then in the early 1900s, two researchers showed that if you cut the tip off of an oat sprout, but then insert a little block of gelatin like agar, the plant will still experience that tropic response. The agar is very permeable. So this told them that maybe it was chemical messengers diffusing down from the tip that caused this. Because if they repeated that, but put a little chip of mica, which does not let water pass through it, the plant loses that tropic response. Then in 1926, Fritz Wendt took this experiment one step further. He cut off the tip of the oat shoot, put that tip on a block of agar, let the chemical messengers diffuse into the block, and then played around with putting that block on different seedlings that had their tips cut off. If you put the block directly on top so that all the cells all around the stem are experiencing it, the plant loses the tropic response. If you put it off center a little bit, you find that the plant bends away from the side where you place the block. And what he thought is that somehow whatever chemical messenger is in that block that came from the tip, it's causing asymmetrical growth. The cells are getting longer on that side, and it causes the stem to curve. We know now that the chemical messenger is a hormone called auxin. There's actually a whole family of auxins in plants that are responsible for lots of particular responses. So phototropism is caused by hormones called auxins that result in asymmetrical growth. the cells on the shaded side are growing faster because of the presence of auxins than the cells on the lighted side. And as those cells elongate, the stem bends and the plant grows toward the light. And we now have an understanding of how that works. The auxins cause hydrogen ion pumps in the membranes to become active. Hydrogen ions are pumped from the cytoplasm of the cell into the cell wall. That lowers the pH. At a lower pH, some of the hydrogen bonds that cross-link the cellulose molecules are broken. That makes the cell wall looser. The internal hydrostatic pressure then causes the cell to stretch and elongate, and that results in that asymmetrical growth. The second way that hormones affect plants is in the overall growth form of a plant. Some species have very strong apical dominance. The apex is the tip, right? So pine trees, for example, have very strong apical dominance. Throughout their life, they retain kind of a triangular shape. And that's caused by the hormone auxin, the same one that results in the phototropism. Auxin at different concentrations have different effects. If you look at the blue side of this graph, in lower concentrations, auxins stimulate growth, but at higher concentrations, they inhibit it. So up and down the stem of a plant, you can have an auxin gradient. If the plant loses its tip, it will often lose that apical dominance. It will become more bushy, right? So people that garden, for example, my wife grows basil, and if she wants the plants to get bushier, she'll pinch off the growing tip, and that causes the plant to lose its apical dominance, and it causes the lateral buds to experience more growth, and the whole plant gets bushier. So remember, flowering plants, the ovary ripens into fruit. And that transformation from unripe to ripe is a hormone-guided process. And one of the principal hormones is ethylene, which can be volatile and be airborne. And ripening fruit can affect neighboring fruit. This is a simple demonstration where there are 3 bananas. Number 3, I guess you'd say, is the control banana. Number 2, the banana is sitting next to an open beaker of ethylene gas, and look how brown it got. Banana number 1 is sitting next to a ripe orange, which is emitting ethylene and inducing that banana to become riper faster. Commercial growers will often pick tomatoes when they're green and expose them to ethylene to accelerate that ripening. In temperate climates like ours, plants often will lose their leaves in the fall. When leaves drop off a plant, that's called abscission. And it is induced by hormones caused by changes in light cycle, temperature, and moisture. There's a hormone called abscisic acid, which induces the formation of a layer of tissue at the petiole base where the leaf attaches to the stem, where it blocks the vascular tissue and causes that leaf to senesce and drop off, and it seals off the end of the xylem and phloem. So not only is the final ripening of fruit hormonally induced, but the actual proliferation of the ovary tissue is under the influence of hormones. There's a group of hormones called gibberellins. Here, this farmer sprayed the bunch of grapes on the right side with gibberellins, and look how big they got. It induced hypertrophy of the ovary cells to make the fruit bigger. There is a specific gibberellin called gibberellic acid that is an important hormone in seed germination. So one of the things that gibberellic acid does is to activate the enzyme that breaks starch down into glucose, making that available to the embryo to allow the shoot and root and cotyledon to grow and the seed to germinate and penetrate the soil. Our final example of responses in plants that are under the influence of chemical messengers is defense. Plants use chemical messengers to defend themselves. And here's a really interesting example. This plant's being chewed on by a caterpillar. So cells are being damaged. There are chemicals in the saliva of the caterpillar that the plant cells are responding to. In response to that, the plant is synthesizing volatile chemicals that go into the air. So here's a chemical messenger that's being released into the air that is attracting wasps that are parasitic on the caterpillars. Those wasps will lay their eggs on the caterpillar, and the larval wasps will actually eat the caterpillar from the inside out while it's still alive. That's pretty gruesome. Our very last slide in this presentation is how plants can use hormones to ramp up their defenses when they are infected by pathogens. So here we're seeing how a fungus affecting leaves in one part of the plant can cause hormones to be produced that travel through the phloem to other parts of the plant, and in response to those hormones, those as yet unaffected parts of the plant might ramp up production of, let's say, maybe the cuticle or other defensive chemicals.